right. Are we chatting? Can you see? Okay. Now, hi, I'm Winnie Linton, and this is my AP Bio first period class. Say hi.
Hi.
Okay, and we are live streaming for 6 life ever began and how certain traits could have been selected for. Okay, now remember, we don't have any, I'm going to show you on a little comedy sketch. And hopefully it doesn't offend anyone.
On Twitter in the past 48 hours, then you've heard about our next story. Rapper B.O.B. blowing up Twitter with his theories on the actual shape of the Earth.
Rapper B.O.B., perhaps you know him for hits like Airplanes and Nothing on You. He firmly believes that the Earth is flat. He's on a Twitter mission to prove it, and he says he has photographic evidence and that we've been fooled all this time.
Hashtag ignorance.
You know, guys, first time I heard Nothing on You, I definitely thought this B.O.B. guy, he is the one to finally take down that asshole Aristotle.
Sorry.
Seriously, though, these tweets are amazing. The horizon is always eye level. Once you go flat, you never go back. Where is the curve? B.O.B., if you're looking for the curve, you gotta call Sir Mix-a-Lot. That is the man who did not deny science. He did not. Am I right? Oh, no. I'm not lying about that. This thing would
Is it still there?
No, it's not. Renowned super scientists in print of the 90s, Neil deGrasse Tyson got involved and refuted some of B.O.B.'s retrograde nonsense, which prompted B.O.B. to release a diss track called Flatline. Here's the tape.
Neil Tyson need to loosen up his vest and probably write that man one hell of a check. Flatline. You got me once, but that died.
Hey.
This just got real, y'all. Uh-oh.
I can't even believe these things happen.
A red science emergency. So I'm going to have to do something I rarely do. I got to hit the science panic button.
Science Emergency Defense Program initiated. Science Emergency Defense Program initiated. Science Emergency Defense Program initiated.
Sandwich. Are you hungry, Luke? Can't believe I'm live streaming on YouTube. On live stream.
I got your distress call. Is everything okay?
No, Neil, everything is not okay. This BS about the Earth being flat is getting out of control. Can you please help us?
Hold my sandwich.
Oh, sure.
Sorry.
Here, you take that. Here. You want to
I literally just said I want to hear it.
Listen, B.O.B., once and for all. The Earth looks flat because, one, you're not far enough away at your size. Two, your size isn't large enough relative to Earth to notice any curvature at all. It's a fundamental fact of calculus and non-Euclidean geometry. Small sections of large curved surfaces will always look flat to little creatures that crawl upon it. But this whole thing is just a symptom of a larger problem. There's a growing anti-intellectual strain in this country.
Yes, there is.
It may be the beginning of the end of our informed
Anti-intellectual strain.
Of course, in a free society, you can and should think whatever you want. If you want to think the world is flat, go right ahead. But if you think the world is flat and you have influence over others, as would successful rappers or even presidential candidates, then being wrong becomes being harmful to the health, the wealth, and the security of our citizenry. Discovery and exploration got us out of the cave.
Really?
Generation benefits for what previous generations have learned.
Anti-intellectualism, anti-science, big time.
If I have seen
For sure.
it's by standing on the shoulders of giants. So that's right, B.O.B. When you stand on the shoulders of those who came before, you might just see far enough to realize the Earth isn't flat. This is called gravity.
All right. So, the only reason I show that video is because people actually talk about it. Like, there are groups of people who actually believe that the earth is flat. Sorry, I do love John Oliver. Okay. So I wanted to start with that. Okay. Now, you know the 3 domains. Oldest BioBuddy. Just give a quick rundown on the 3 domains. Go ahead. Are those showing up? Yeah, they are. What do these 2 have in common right here? Prokaryotic. And this is, what might separate bacteria from archaea? Extremophiles. Good. All right. Now, youngest BioBuddy, we have already talked about Luca several times, but just go ahead, youngest BioBuddy, give him the rundown on Luca. Here you go. Now, what are some of the pieces of evidence that we've already discussed that support the idea of Luca? Fossil evidence? Okay, what else? Okay, those are all evidence for evolution. But specifically, like, can you think of things like, yeah.
Like the ribosomes.
Yep, the ribosomes for protein synthesis. What?
The Stark codon.
Stark codon, AUG, you got it?
The amino acids in cytochrome C.
Yes, the amino acids in cytochrome C, the universalness of, like, what's something that everybody does? Cellular respiration, right? The pathways in that. That the introns that get cut out are similar, right? How is the stuff that gets cut out that's not even expressed? Why is that similar? Unless it came from a common ancestor, right? Because you're not expressing the introns. Metabolic pathways, genetic code, universal, the structure of the cell. So these are all things that point to a universal common ancestor. So when we talk about the origin of life, we're going to look at 2 big phases. Phase 1 is just getting us to the beginnings of a cell. And so this is all chemical evolution. Evolution of chemicals. We're trying to get to the point where we have our first living thing. So we need to have the monomers. Do you remember the 4 important monomers that we learned about? Or sorry, let me make it easier. Let's start with polymers. Monomers build polymers. What are the 4 important organic molecules that we talked about? Carbohydrates, proteins, nucleic acids, lipids. Okay, so those are the polymers. That's step 2. Step 1 is what builds each of those polymers. So carbohydrates are built out of what? Sugars. So you got to have sugar. Did the early Earth have sugar? No. So how did that come about? That's what we're going to talk about today. What are proteins built out of? Amino acids. Good. What are lipids built out of? There's 2 categories. What are the 2 categories of lipids? Fats and steroids. Fats are built out of what 2 things? Glycerol and fatty acid chains. And steroids are built out of what?
4 carbon rings.
4 carbon rings. Good. And nucleic acids are built out of nucleotides. What are nucleotides built out of? 3 things. Base, sugar, phosphate. And you were saying nitrogenous. Yes, nitrogenous base. Okay, those are the monomers. So first we have to get the monomers so we can build the polymers. Once we have the polymers, we could build our cell. For instance, a cell membrane is composed primarily of what 2 things? Phospholipids and proteins, right? The fluid mosaic model, you know all of that. Dig deep, okay? So 2 of the 4 important organic molecules are in a cell membrane, but we found others there too. What other one did we find in a cell membrane? Cholesterol, which is a lipid. Where else? Not in plant cells, but yes, in animal cells. What else? Glycolipids and glycoproteins, which are carbohydrate side chains, right? So 3 of the 4 important organic molecules are found in just a cell membrane. So you need to have these polymers to begin your first cell, your beginning of your cell. You're not a true cell yet when you're a protocell. So that's chemical evolution. Step 2 is biological evolution. Once you become self-replicating, you're a cell that makes more cells. And by the way, what do you think the first cell was, a prokaryotic or a eukaryotic cell? Prokaryotic cell. Once you have your first cell that's prokaryotic, then do you think the first cell was an autotroph or a heterotroph? What do you think is simpler? Go out and consume nutrients or make the nutrients that you're going to put through cellular respiration? Consume. So would you be an autotroph or a heterotroph? Heterotroph, just consuming nutrients. So being a heterotroph more than likely came first and then an autotroph. Okay, remember when we did photophosphorylation, right? Remember there was cyclic and non-cyclic photophosphorylation. You see how this is bringing together all of your past knowledge? So think about it, right? Just tell your bio buddy, what was the difference between cyclic and non-cyclic photophosphorylation? Go ahead. Okay, so let me jump in. Cyclic photophosphorylation. You did not need a source of electrons. You didn't have to keep supplying the source of electrons, right? Those same electrons got excited, and you make some ATP. Get excited, make some ATP, right? What didn't you make as a result of that in cyclic photophosphorylation? You didn't make any what? Reduced NADP, right? But when non-cyclic photophosphorylation evolved, which is during the what revolution? Oxygen revolution, about how long ago? 2.3 billion years ago. Then what molecule got split? Water. And as a result of that, you got oxygen and a source of your electrons, right? As you split the water, because the electrons came from where? From the water, but for what part of it? Hydrogen. Those electrons got excited, make some ATP, excited, and use it to reduce some NADP, yes? So when you walk away with ATP and reduced NADP to do the dark reaction or the exactly. So you remember all of that, yes? Okay, so self-replication, heterotrophs, autotrophs, and then eventually you would go from unicellular to multicellular, okay? All right, good. So that's the big picture of where we're going on all of that. Is that all transmitting well? Is anybody looking on YouTube? Is it going across okay?
Yeah.
Okay,
It's
sweet.
a little blurry.
It's a little bit.
I don't know if it's my connection or.
The connection of it, yeah. Or I think when we record it and then we stop it, that when you have time to catch up with the streaming, I think it comes across sharper. Maybe. Let me know if I'm doing something wrong. Can you guys see if somebody puts a message up there? Do you see a comment? So just tell me if there's something I need to address. Because I can't see it because it's too far away and I'd have to have my glasses. All right, so let's start a long, long time ago. So if you look down here, the universe is about how many billion years old? 14. Okay, good. And then the Earth is about 4.6 billion years old. Okay, uninhabitable. You've got molten magma going. You have continents shifting, right? You've got the very first atmosphere we don't even really talk about. The Earth did not have the density that it needed in able to hang on to its first atmosphere. That was gone. The second atmosphere probably is one that came out of volcanoes, outgassing of volcanoes. The good thing about where we live in distance, the Earth distance to the sun, it's like when I get out of the picture, then the smartboard gets better. The distance from the sun allows water to live in all 3 states, right? What are the 3 states? Solid, liquid, and gas. So as the Earth cooled and the gases cooled, then you had oceans. And the gases of the environment were probably those that just came out of a volcano, right? So how much of the Earth is covered in water currently? 75%, right? And then you have these continents. You have gases coming out of the volcano. The gases that came out of those volcanoes on that early Earth, they were probably, it was a very reducing environment. Do you remember the difference between oxidized and reduced? Oldest bio buddy, take the lead on that. Remind them, what does it mean to be oxidized and reduced? Okay, when you become reduced, you gain what? Hydrogens, you gain electrons, you gain energy, right? Is it about building or breaking? Building. When you oxidize something, what are you doing? Breaking it down, right? If I took a key to your brand new car and went along your paint job, the oxygen in the air would interact, right, with the metal of your car and start to break it down, oxidize it, right? Okay, the early Earth, the gases that were in the environment in the early Earth, can you see anything that's missing that is in the Earth now? Oxygen. Now, there is oxygen like carbon monoxide. You have oxygen H2O, but you don't have any free oxygen as O2. This is critical because it was conducive to building, not breaking. Our Earth right now has a ton of oxygen in it, right? Or 22% of the gases that we breathe. There is a lot of oxygen in there, but it wasn't always like that. So molecules could build upon one another, not now. Now they break down. And if you don't see life re-evolving all over again in a puddle somewhere in your backyard because it's not a reducing environment anymore, it's an oxidizing environment, okay? Now, Earth, I already told you that. Earth's distance from the sun allows water to exist in all three stages. So can you see, I tried to use an analogy of stairs. Can you see the stairs in the background, the stair steps? Okay, all right. So in order for us to get, this is a step, 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 step. Do you see it? Okay. So the early gases, the early Earth gases, and I'll help you with your notes in just a minute. Early Earth gases would be similar to what came out of a volcano. It would be these items right here. So the first thing we need to do is we need to get to our bricks, right? We need to get amino acids. We need to get some sugars. So how could that have happened? So we need to go from inorganic molecules to organic molecules. What organic molecule do I have on that step right there? It's an amino acid. Youngest bio buddy, is it your turn? Tell them the parts of an amino acid. You should be remembering this.
Oh, then jump right up there, and then you have separate carbon there, and hydrogen negative.
So did they say an amino group? An amine group right here? Did they say a carboxyl? Did they say a hydrogen, all bound to a central carbon? And then an R group, how many different R groups do we have? 20 different R groups. Perfect. Okay, so we need to get from this point to this point, to this organic molecule, all of these organic molecules. That's the step we need to make, and I'm going to talk to you about that. Then we need to go from these organic monomers to the polymers. So our polymers, we talked about that, proteins, carbs, lipids, nucleic acids. And then once we have these molecules, we can build our first cell. And that first cell would be a protocell. It wouldn't even be a true cell. It's not until it's self-replicating. And probably that first cell was an anaerobic prokaryotic heterotroph. All right? So let's get a few notes down. Grab your notes on the origin of life. Luca, I bet you already did that. What is Luca? Last universal common ancestor. Perfect. Early Earth consisted of what kind of molecules did we have? Inorganic molecules, but living organisms consist of biomolecules that are organic. We will discuss 4 stages hypothesized for life to start. Stages 1 through 3, what kind of evolution is that? Chemical evolution, monomers to polymers to protocells. And stage 4 is what kind of evolution? Biological evolution. Abiogenesis. Can you break down that word for me, please? A means against, right? Against. Bio. So against life. Genesis means what? Beginning of life. So it's life now we know, like we know the cell theory. All living things come from cells, right? Cells come from other cells. But early Earth, there were no cells. So how did life come about without any life there to guide it? For instance, what makes our reactions go faster? Enzymes. Enzymes are made out of what? Proteins. How can you build these things if you have no enzymes to build them? Do you see the problem there? How can you get there? How can you build a protein if you have no proteins? Chicken, the egg kind of concept? Okay. All right. So that part, that's the abiogenesis, the origin of life from non-living matter. Okay, so if you look at your notes, do you see how I've broken it down? Stage 1, stage 2, stage 3, stage 4? Got that? Okay, very concrete, sequential. All right, so let's go with the first one. Let's talk about monomer evolution. So these are the bricks, right? So how do we go from inorganic to organic? This is part of your chemical evolution, this increasing in complexity. So there are several different ideas of how we could have gotten monomers, and I'll talk you through. One could be an extraterrestrial, like a comet. I don't mean like an alien coming and bringing it. I mean like a comet, an asteroid that has organic material on it, landing on our Earth and seeding our Earth with organic molecules. Energy from the atmosphere. What this is meaning is that all the gases that, first of all, make an early Earth. Sorry, I'm going to teach this right. Make an early Earth. Okay. What's going on this early Earth? You have water. What do you think this means? Rain. And you're forming what? Oceans. You have gases coming out of what? Volcanoes, but there's no oxygen. Okay. So just show me again. Early Earth, okay, you have rain that's creating oceans. You have gases out of volcanoes, but there's no oxygen, right? You have all the gases that come out, you kind of like have this soup of all those gases that are going to be mixing with the ocean. We have sources of energy. We have UV radiation. Say it. UV radiation. You have what do you think that is? Lightning. You have lightning. You have molten magma. Okay? And you have radiation from the Earth's core. Okay, for the beginning. Early Earth. You have rain, forms oceans, gases coming out of volcanoes, but there's no oxygen. But you have sources of energy. You have UV radiation. You have lightning. You have molten magma, and you have radiation from the Earth's core. Remember how you learned about that last year in the core and the molecules, right? Yeah, okay. Good. So this energy could have taken those simple inorganic molecules and using that energy formed organic molecules. And then a third one is nitrogen would be, nitrogen in a form that can be used as ammonia would be in short supply. The reservoir for nitrogen, breathe in, breathe out. 78% of what you were breathing in right there was inert nitrogen gas, N2. Your muscles didn't get bigger, you didn't get stronger, your stomach didn't get full. Because we can't do anything with it, neither can plants. Okay, so we would have run out of NH3 ammonia, and that's possible that that could have been formed at hydrothermal vents. So I'm going to show you that right now. Okay, so here's your early Earth. And oldest bio buddy, this is you. Go. I already explained this. It's got pictures. Go.
Europe could have twice.
Okay, now, a man named Operin proposed this. Okay, a man named Miller did an experiment about this. He took what the gases are. You know, people have some different ideas about that, but he built this apparatus. He put the gases in this apparatus to, can I zoom in on that so somebody could see that? Can you do that?
Maybe.
Yeah. Oh, good job. Thank you. Adjust it so you can see it better.
Okay. Just like move it back a bit.
I can't see the top. Okay, so these gases right here from early Earth, now he put this here, there's an electrode. What would the electrode represent? Lightning, good. Okay, then you see the gases down here. There's some sediment, and then what would this be like? What would this heat be? It could be radiation, it could be molten magma, it could be UV creating that heat, and so then they would evaporate and go around and go around. Within about a week's time, he went to check. So he started with the gases like what came out of a volcano. He just added heat here and electric shock here. And he got all of these things in a week's time. A week, okay? So when you put that forth as a hypothesis, how could that, what do we need to just?
I can't see the top of that.
Oh, caca, I got it. Oh, I just said caca. Sorry. I'm sorry. I'm sorry. Can we, is there a tightening thing right here? No.
Oh, yeah,
Okay.
yeah, that's.
Is that better?
It was, but then now it's looking down. Yeah, that's good.
How about now?
It's a 4-second delay, so we'll be able to.
Okay.
There we go.
I
I
can.
think we're good right
Are we good now?
Yeah, right.
Okay.
Yeah, that's good.
Okay. And so within a week's time, now that's like pretty amazing, right? Because if you think about it, how old is the Earth? 4.6 billion years. Okay? So the first living thing, like where we have a fossil, is about 3.6 billion years. So how many billion of years went by? 1 billion years. Going from the formation of the Earth, 1 billion years from the formation of the Earth until the first fossil, first cell. Okay, so you saw the steps we have to go up, right? First step is inorganic to organic. He did that part in a week. Okay, so that gives me the idea that there's a possibility that all of this could have come about if that hard part of just making organic molecules happened in a week's time. Okay, and just in case you're curious, here are some scientists. Okay, I want to bring them up. I know, they're always old white men, like our presidential candidates. Okay, so evolution of monomers. Operon Haldane hypothesis, primordial soup hypothesis. Monomers built abiotically. What does that mean, abiotically? Without what? Without life. From inorganic molecules present. With the energy present. And then I gave you a bunch of energy options. Yes, you have all those? In the environment present. And it was a reducing environment, no free oxygen. Remember the significance of no free oxygen cannot be underestimated, okay? Because if oxygen was there, as soon as these things were built, they would be broken back down. They'd get oxidized. But there was no oxygen to do any oxidizing, so it was conducive to building. The primordial soup hypothesis and the Miller-Urey experiment, early Earth gases, and I gave them to you, were put in a cycling apparatus. Electric sparks provided energy, like lightning, and within a week, all 22 amino acids were formed. Some modifications of the other 20 were formed in addition to other molecules. They're right here. Further studies hypothesized, I'm getting on the nitrogen part, but deep ocean thermal vents, hydrothermal vents, could convert it to ammonia. So let me show you that, okay? Because you can see you on this right here, you need nucleotides. Nucleotides are base, sugar, and phosphate. The base is what kind of base? Nitrogenous base, right? So you need a source of nitrogen that is not N2, okay? That is not bound as a diatomic molecule. So where could that have come from? These deep hydrothermal vents could provide the energy, the catalyst in order to, it's somewhere here, good. These iron nickel sulfides in order to convert nitrogen into ammonia, so you would have a source of ammonia. Okay, so on your notes, you can also see iron sulfur world hypothesis is number 3. And Slate's going to take that on because you have everything. And Blue's going to take on extraterrestrial origins hypothesis. Go ahead.
Thermal vents. Okay, thermal vents.
Case. Oh, let them listen to you talk loud.
Thermal defense could have provided all the necessary ingredients to form monomers. That's the iron sulfur world hypothesis. And then the extraterrestrial origins hypothesis is meteorites could have seeded the Earth with organic molecules or even cells.
Okay, is that a better view where you can see the screen kind of viewed in and I just step in occasionally? Is that better?
Yeah.
Okay, good. Yeah, because it was too far away. All right, now, next, we've gone up our first step and we're standing on our first step. We have some more steps to go. We're still part of the chemical evolution. So now we've got to get the monomers to be the what? Polymers. But we're doing this because we don't have any ribosomes to build the proteins on, right? We don't have any fatty, we don't have any smooth or rough ER and how to make us more fats, right? We don't have a way to replicate the genetic code. We don't have any DNA polymerase. We don't have any RNA polymerase. How are we going to make more of this? So polymer evolution is where you take those bricks, right, to build your wall. And the big question is, what came first? The code. By the code, I mean a nucleic acid like DNA or RNA, or the enzyme that you need to build the code. But the code codes for the protein, which is the enzyme, following the conundrum there, right? So what came first, proteins, RNA, or both? So, oh, that should say proteinoids. So there's a protein first hypothesis, there is an RNA first hypothesis, and there's a same time hypothesis. So the key thing you're wanting to form is you're looking at those sorry, this whole slide got jacked. I just apologize. Is how you're going to form those nucleic acids and those proteins. So first off, the monomers that you have, you could have the same energy source provide the energy for you to build those polymers. What were all of our energy sources? UV radiation. What? Lightning, magma, radiation from the Earth's core. You still all have those to provide energy, and that could facilitate it. You could concentrate molecules because as water would evaporate out of puddles, it would concentrate those molecules more likely to react. I'm just jumping to several of them right now. Clay can act and does now act as a catalyst. Like, remember, an enzyme is an organic catalyst, right? So can act as a catalyst, the surface of clay, to facilitate reactions. So that could have done that as well. There's a man named Fox. I thought his name is Sidney Fox. Sorry, his X is gone. He kind of looks like 1970s movie producer, kind of slimy to me, but I'm sure he's a very nice man. I don't want to, but I just thought I'd show you what he looks like. His whole idea was, see, so this is to help you remember, Fox, okay? Sidney Fox, his idea is that proteins came first, that they came first. And the reason is when you could concentrate them, you have energy from the sun, amino acids could come together to form proteins, but also his idea extends not just to the polymers, but his idea extends all the way out to the first cell, because remember our cell membranes are made out of what two molecules? Lipids and proteins. So not only does he say your first polymer would be like a protein, but your first cell membrane would be protein-based. And he says this because when you take hot proteins and you cool them quickly in water, they will form a sphere shape. And they are actually called Fox's proteinoid microspheres. And some scientists have actually been tricked by them and thought that they had discovered a new life form. But they were, because they would like kind of line up like staphylococcus, like these spheres all hooked together, but they're just literally cooled proteins in forming this sphere shape. So he does it for polymers and also the first cell. So on your notes, go to stage 2, evolution of polymers, without the assistance of enzymes that are polymers. Slate, take care of that iron sulfur world again. You've done it before. Do it again. Do number 1 right there.
Iron nickel sulfides with their charged surfaces could attract and bind amino acids together.
Okay, and then the protein first hypothesis from Fox, heat from the sun could form proteinoids, small polypeptides that have some catalytic properties, which then could be placed in water and form microspheres, which would then be the early cells. Okay, good. And then next, the RNA first hypothesis. Now, Thomas Cech and Sidney Altman shared a Nobel Prize in 1989 for this. And that is because RNA can act as a code, genetic code, right? Because we know we take the central dogma says we use the DNA and we form RNA from it. What kind of RNA? mRNA, tRNA, and RNA. But mRNA is the messenger, right? And then we use that mRNA to build what? Proteins, right? So RNA is your genetic code. Maybe the RNA came first. And because RNA can also act as enzymes. Remember, the ribosome is enzymatic in catalyzing the joining of two amino acids together, yes? So if RNA came first, it could facilitate that process just like a ribosome does. Also remember spliceosomes? Remember, cut out the introns before it leaves the nucleus? Okay, remember SNRPs? Made out of spliceosomes. So it can act enzymatic in cutting and manipulating the nucleic acid. So maybe RNA came first. Remember, I've talked to you a little bit about this in that RNA could be first and DNA just evolved as a way to store the RNA. Does that make sense? Okay, DNA evolved as a way to store it. So that's what they believe, the RNA first hypothesis. Then, okay, so on your notes, go to RNA first hypothesis. RNA could be a substrate and an enzyme. A substrate and an enzyme. Okay, and then the first cell. So we have a protocell is defined as a lipid protein membrane, and it carries on energy metabolism. It's breaking down molecules in order to make ATP, some source of energy, right? So Fox would say that first cell, he would go with those proteinoid microspheres. Operin, he had an idea called coacervate droplets that, and if you can think about it like this, is the cell membrane consists primarily of phospholipids or proteins. Fox goes the protein route, Operin goes the fat route. And he says it's like fat droplets. And that could have formed that first initial barrier. So whatever is going on inside the cell is different than what's going on outside the cell. Okay, so that area could evolve. Autotrophs, heterotrophs, what did we discuss is probably most likely to have occurred? Heterotrophs before autotrophs because it's simpler. Okay, not it. One of you is going to take this slide, and the other one, well, actually, why doesn't one of you take protocell membrane, and one of you compare that to the cell membrane? Go ahead.
Protocell membrane does have a tail, but it only has one so it's one individual fatty acid. There's only one.
And that makes the phosphorus
Okay, sorry, I'm back. Good. All right, so here are Fox's proteinoid microspheres. They look very much like, a prokaryotic cell. And then here are Operin's coacervate droplets, and these are just droplets of fat. It's probably a mixture of both of those. It could have happened maybe at the same time or similar. Who knows? These are just hypotheses that are put forward. What they do have to have is they have to have some sort of metabolism, and probably the first one you already told me would be a heterotroph, not an autotroph. But of the heterotrophs, what are the two types of respiration? Anaerobic and aerobic. So probably what evolved first? Anaerobic. Why? Because there was no oxygen, not till after the oxygen revolution. So you probably had prokaryotic heterotrophs, then what? Prokaryotic autotrophs that were doing what? Well, of course they were doing anaerobic respiration, but what else? Cyclic, non-cyclic, which one? Cyclic. Cyclic photophosphorylation. Okay, let's think it through again. First cell, prokaryote or eukaryote? Prokaryote. So prokaryote and having to consume, so that'd be a what? Heterotroph. Okay, what kind of metabolism did this heterotroph have? Anaerobic respiration. We all get with that? Who could have evolved next? Prokaryote, autotroph, doing what? Cyclic photophosphorylation, yes? Then who could have evolved next? Prokaryote, autotroph, non-cyclic, creating the oxygen revolution. So now we could have prokaryote heterotrophs doing aerobic respiration. Then we could evolve from being a prokaryote to a eukaryote, right? Then we could go from being unicellular to multicellular. There you go. Okay? Putting it together logically, right, through that path. All right, so on your notes, plasma membrane, a boundary was required for regulation and maintenance of cellular activities. And Slate, finish the rest of that sentence starting if.
Lipids are made available to microspheres. Lipids are made available to microspheres. They form a lipid or protein membrane. Micelles, fatty acids, coacervate droplets, liposomes, and lipids.
All right. Next, nutrition, probably anaerobic fermenters, only participating in glycolysis. Anaerobic fermenters, only participating in glycolysis. All right, now we need to go up to the next one. Okay, so did RNA first, protein first, same, in order when we were self-replicating a true cell. And there are basically three ideas here, of which we've already actually discussed. We talked about the RNA first hypothesis, right? Because it can act as both a substrate and a enzyme, okay? Sidney Fox, same proteins first, and your first cell membrane would be like a cool, hot protein cooled into a sphere. Then this one is new, clay. And the idea here with clay, and let me just get, here we go. The idea here with clay is that RNA and proteins formed at the same time. And they did this because clay contains iron and zinc that catalyzes reactions that can facilitate the polymerization to form both proteins and to form nucleic acids. Because what we're trying to get to is we're trying to get to where you're in your self-replication, where you're making more of yourself. So that means you need more nucleic acids, you need more proteins. I want to remind you of the central dogma. And what this would be saying is that you started this way at RNA, and then we went back to store it as DNA and went forward to build it as proteins. Does that make sense? That's the RNA first. Okay? Karen Smith on clay says these and these evolved at the same time with the use of clay as a catalyst. But at the point where you can make more of these things and more cells, then you are a true cell. So evolution of the genetic code, not it? Pass or play, go.
CCG values or CCG codes
And this is evidence for what? Evidence for what? Luca. Good. Okay. So then your first fossil is about 3.6 billion years old, and that's probably a prokaryotic heterotroph. Then later autotrophs and eukaryotic cells. So on your notes on stage 4, evolution of the self-replication system, RNA first, RNA coded for proteins, then hypothesized that reverse transcriptase formed DNA as a storage location for the genetic code. Protein first, RNA is complicated. More likely that proteins evolved first to guide the synthesis of nucleic acids. Protein RNA simultaneously. First cell would have true genes that could have been replicated because of the presence of proteins. And the genetic code probably underwent natural selection to efficiently store that information. Underwent natural selection to efficiently store that information. Best out of 3, rock, paper, scissors. Go. Okay, whoever is the rock master, pass or play, started from the bottom, now we're here. Okay, go ahead. All right, let's go in today as our favorite color in the crayon box. You should use all the colors in your crayon box. Favorite color in the crayon box. I'm waiting for you to get all your names out of there. And if you want to go in at home with us, my face is huge. If you want to go in at home, you just go to hellosmart.com, hellosmart.com, and the code is 878. Here, I'll make it bigger. Here's your code. And then you can join us at home. Go in as your favorite color in the crayon box.
I wish I just
Good Night Moon. That's a color in the crayon box? That's a great book, but I don't know if it's a color in the crayon box. Are there more of you trying to get in? Because there's more than 22 of us here. That is a good color. All right. Is there somebody struggling to get in? Raise your hand if you're struggling.
There's 24.
Oh.
I remember that. It was my favorite. That was my favorite. Green.
Anybody see where I put my phone down? Oh, I'm so stupid.
My life does not.
Where can I have that?
We didn't talk about it.
Stupid. Anybody know what my phone is? I don't know. Is it the thing sitting in the middle of the classroom?
Sorry.
Maybe. Maybe, maybe not.
Have you ever like been on the phone?
Yeah, sure. I have at times have had 2 pairs of glasses in my hair at the same time.
How did you know?
I don't know.
The answers are. But that's the, you'll see.
Let's go cerulean. That's a blue. Let's go existential red black. Let's go mermaid.
Existential
Person.
red black.
Existential red black. All right, here we
You
go.
have the last one. Just wanna let you know. Ahead of time.
Are you sure?
Yeah, totally.
The atmosphere in which life arose lacked the source of free oxygen. Really? Nitrogen? Come on. See if that was your bio buddy. Have words. Number 2, why doesn't life not arise today? There is not a reducing atmosphere today. What kind of atmosphere do we have today? An oxidizing atmosphere. It's not because there's too much pollution. Seriously, I went over this. It's terrifying to me. Number 3, scientists who performed an experiment that supported Operan's hypothesis, Stanley Miller. Graham's Carn Smith was the clay for proteins and nucleic acids at the same time. Sidney Fox was the protenoid microspheres. Okay. Protocells probably obtain energy as heterotrophs, not chemoautotrophs. They would be chemoheterotrophs, but they were not chemoautotrophs, right? Autotrophs means you have the ability to take inorganic carbon dioxide and form organic sugar. They did not have that ability. Okay, number 5, prokaryotic cells are represented by fossils that are as old as 3.5. So 3.5, 3.6, those people that pick D, what are they talking about?
Earth.
This is closest to the oxygen revolution, 2.5. 1.5, closer to 1.7, is actually the beginnings of forming eukaryotic cells, the beginnings of it. All right, so now we are at, we're going to do our last 6 minutes hard as far as we possibly can get. And then I want to talk about the coronavirus. Okay, so fossils. Have we talked about fossils already? And I passed them around once, right? So we're familiar with fossils. So that's easy. The topic says fossils tell a story. The majority of fossils are found in sedimentary rock. Now, the assumption is that the layers on top are what? Newer or older? They're newer. And the layers on the bottom are? Yeah. Now, you have to look for convergent boundaries where one layer might go underneath another set. So you have to be able to analyze what's going on with the plates at that point. Now, for perspective, I don't think, have I shown you this picture? Okay, so let's do this for perspective, like all time on the earth since the beginning of time. Okay, if you look at midnight last night, which I hope you were asleep in bed. I don't know what you were doing at midnight. You're like, no, I was doing homework because I was at mock trial. Okay. So, midnight last night, imagine that as being the formation of the earth. Midnight last night. It's 1, it's 2, it's 3, it's 4 in the morning. We are in the Precambrian period. Okay? 5. Oldest fossil prokaryotes were at 5 in the morning. Miss Litton was just finishing exercising at 5 this morning. Okay? So oldest fossils are prokaryotes. Okay? Now look right here. At 6 in the morning, okay, done with coffee, getting a shower, first photosynthetic organisms right here. Okay? Still in the Precambrian period. It's 7, it's 8, it's 9, it's 10, which is right now. Okay? It's 11, we're still in Precambrian, 12, 1 o'clock this afternoon is when oxygen would show up on the scene. Oxygen revolution. 1 o'clock this afternoon. Still in Precambrian period. 2, 3, 4, look, oldest what? Eukaryotic fossils after school today at 4. Remember, we've been running this clock since midnight last night. Still in the Precambrian time period. It's 5, it's dinner time, it's 7, it's 8, it's between 8 and 9 before we even had multicellular or multicellularity evolving. Okay, 8 and 9 tonight. At 10 tonight, we enter the Paleozoic period. This is when you have land plants, okay, and lots of things happen there. If you look right here, Mesozoic, we're like at 11 p.m. at Mesozoic. You know Mesozoic because the middle period is called the Jurassic period. What happened then? Yeah, so dinosaurs were a big deal in the Mesozoic. Look at this. It is Cenozoic, getting closer to midnight at 11:59 and 30 seconds. The last 30 seconds of our 24-hour clock, who shows up? Man. Okay? I do that so you see the time and the perspective, okay, of what has occurred and how long it took everything to occur, okay? Now, on your notes, you have 24-hour Earth diagram illustrates that only single-celled organisms were present during 80% of the Earth's time. 12 midnight, Earth forms. 5 a.m., first cell. And what was that first cell? 4 p.m. we had eukaryotes. 8 p.m. multicellularity. 10 p.m. invasion of land. 11:59:30, who showed up? Yeah, humans. Okay, besides sedimentary rock, you can find fossils in amber, tar, ice. There's some amber for you. Here's some ice. Here is some petrified salmon. We already learned about molds and casts. Remember when I brought out my fossils from home? Okay, so those are all different kinds of fossils. Here's a fish. That's an imprint. Here's another one. Okay, look at how big he is. It's ginormous, right? Now, there are two main types of dating, relative and absolute. Relative is when you can't find anybody else to go to prom with and you ask a cousin. Kidding. And then absolute dating is you're getting an actual number. Like relative dating, you're going to say that these layers are newer than the layers below it, and so you're using that, okay? Absolute dating is when you're looking to see how fast an isotope breaks down, and you had a whole big section in HyperDoc on that last year, so I know you know about that, right? Remember we talked about what kind of lives? Half-lives. The time it takes for half of whatever you have to break down. So when you look at ratios and radioactive dating, that's absolute dating. You're actually coming up with the time. Okay, now one thing that helps in that process are index fossils. Index fossils have been found again and again only to occur at a certain time period. So if you go find a rock layer and the index fossil is beneath that, then you know everything above that is younger. If you find layers below that index fossil, you know everything is older. So it's combinations of those two things. Also, you can match. You can combine them both, but we can match. Remember when we looked at Pangea, like and how the continents fit together? Yes. Yes, thank you, because I would fight you on that. While I get the video, I'm going to freeze it right here and not it. Talk about absolute dating, and we will pick up from here next time. It was so glad to have you, and I hope you're having a great day. We're going to watch on ASAP Science. It's on the coronavirus. It's a really pretty good video. It's about 8 minutes. I highly encourage it. Yes, I do.